Hello again, everybody. Thanks to everybody who's attended so far to attend this webinar on COVID-19 virus variants, and in particular on the topic of the emergence of new SARS coronavirus 2 variants and their resistance to antibody neutralization. While we wait for a couple more attendees to join, I will take this opportunity to introduce the presenter. Today we have Dr. Clark Shear presenting for us. Clark has two decades of experience in drug discovery and development, including work in a number of Canadian pharmaceutical companies on various therapeutic programs. Clark received his PhD in biochemistry and molecular biology from the Shanghai Institute of Biochemistry. And then he followed up with his postdoctoral training in the Department of Biochemistry at the University of Washington in Seattle. He has since worked hard to become an expert in developing various biological assays for target discovery and validation, and is currently the director of the molecular biology at Signalchem. Signalchem, his place of work, was founded in 2004, located in Richmond, BC, Canada, and is a global leader in protein engineering, manufacturing, purification, and assay development. With that said, I would like to pass this virtual mic over to Clark to begin his presentation.
Thank you very much, Paolo, for your introduction. And good morning, everyone. I'm glad to be here this morning, and thank you for joining me with my discussion on the emergence of SARS-CoV-2 variants and its impact on the treatment and prevention of COVID-19. First, I will give you a very brief background introduction of coronavirus and COVID-19. Then I will introducing the emerging SARS-CoV-2 variants. Many variants have been identified since the outbreak of COVID-19. Today, I will only focus on several variants that raised great concern because of their increased transmissibility and potential immune escape to antibodies. After that, I will talk about the development of COVID-19 vaccines, different types of vaccines, and their effectiveness. At the last part of my presentation, I will discuss the resistance of the new variants to antibody neutralization and its clinical consequence. As we all know, the COVID-19 pandemic has led to a dramatic loss of human life worldwide and present an unprecedented challenge to public health, food systems, and the world of work. The economic and social disruption caused by pandemic is devastating. Since the outbreak of COVID-19, more than 127 million cases have been reported globally. Among them, close to 2.78 million people have died from this disease. At the bottom right, there's a chart showing the daily COVID-19 reported. Starting from late February last year, the number is continuously growing. It accelerated in fall from October and picked up after the Thanksgiving and the Christmas season. Then we start to see the reduction of the number. I believe this is due to the mass vaccination program took place in countries like US, UK, and Israel. It is very promising to see that. But I am very concerned when I see the numbers start to rise again, starting from February this year. Why did that happen? Will the rising continue? I don't know. We need to keep watching on it. Now many governments are trying to reopen their economy. Given seeing the turnaround of the number of daily cases, I think it is better to do it slowly and carefully to avoid the third wave of a pandemic. COVID-19 is the name of the disease caused by novel coronavirus SARS-CoV-2. Coronavirus are enveloped RNA virus. That means that they have RNA as their genetic materials surrounded by the fatty layer skin. Coronavirus can be classified into three groups: alpha, beta, gamma. Current SARS-CoV-2 belongs to the beta group. Coronavirus can primarily infect birds and mammals, include human beings. That means they can cause a cross-infection between people and animals. Recent pandemic caused by the coronavirus, SARS in 2003, MERS in 2012, and the current COVID-19 emerged in late 2019. Here's the diagram showing the coronavirus particle and the genome structure. Inside the particle, there's a single-strand RNA associated with a nuclear capsid protein called N protein. SARS-CoV-2 has a positive RNA strand encoding a small non-structured proteins that are very important for viral replication. And structured proteins such as spike protein, membrane protein, and envelope protein, those are involved in the viral contacting with host cells and leading to the entry of virus into the cells. SARS-CoV-2 contacted and adhered to the host cells through its spike protein binding to the receptor on cell surface, which is ACE2. The angiotensin converting enzyme 2. After binding, the spike protein is processed by protease like membrane-associated TMPR SS2 showing here, and the furin. The cleavage causes conformational change of a spike protein and leads to virus fusing with cell membrane and gain entry into the cells. Recent studies showed that other membrane receptors like AXL, I show on this slide, also play a role in the viral entry into the cells. Blocking or inactivating AXL can inhibit the infection. COVID-19 first emerged in Wuhan in China in late 2019. It was announced to be a pandemic by WHO on March 12, 2020. The most common symptoms are respiratory symptoms. Cases can be infectious with or even without symptoms. Majority of the cases are milder. In the most severe cases, people can develop difficulty breathing and may ultimately experience organ failure. Some cases are fatal, but deaths have generally occurred in people with underlying health conditions. Now let's talk about the SARS-CoV-2 variants. The first variant that brought great attention is this one, D614G. The variant has a mutation of aspartic acid to glycine in its spike protein. This variant emerged in late January or early February last year. It quickly became the dominant form of the virus circulating globally. Actually, the variants of concern I'm going to discuss later all have this D614G mutation. This variant has increased in infectivity and transmissibility compared to the original virus strain. Fortunately, this variant does not cause more severe illness or alter the effectiveness of the existing laboratory diagnostics, therapeutics, and vaccines. Then we have the UK variant, the variant with higher infectivity and transmissibility. This variant was first found in September 2020. Initially appeared in southeast England. Just within a few weeks, it began to replace other virus strains in that area and in the city of London. The variant has increased infectivity and transmissibility. It is between 40 to 70% more infectious than the original strain. Early this year, the scientists from UK published their study results, reporting the evidence that suggests this variant may be associated with increased deaths compared to other strains. More studies are needed to confirm this finding. The UK variant has 17 mutations. The mutations do not randomly spread across the genome. Rather than clustering in its spike protein, 9 of 17 mutations are located in spike protein. Notably, a mutation, the N501Y, is located at a key position for RBD interacting with ACE2. This mutation was shown to enhance the binding affinity of RBD. At Signalchem, we produced the recombinant RBD with N501Y mutation and tested its binding with soluble ACE2. You can clearly see here the mutation increased RBD binding affinity to ACE2. Mutation P681H is located right next to the fusion cleavage site. Evidence suggested that it may be associated with higher infectivity and transmissibility of this variant. The deletion of 69 valine and 70 histidine residues has been detected in multiple independent circulating lineages of SARS-CoV-2 and is linked to immune escape in immunocompromised patients and enhanced infectivity in vitro study. This deletion also caused certain commercial diagnostic assay failing to detect the spike protein gene. Then we have the worrisome South Africa variant. This was found in the beginning of November last year. At the time, it has largely replaced other strains in 3 provinces in South Africa. Again, you can see the mutations are clustering in spike protein. It also has D614G and N501Y mutations that are found in the UK variant. So this variant is also named 501Y-V2. Besides the N501Y, it has 2 more mutations in the RBD, K417N and E4A4K. Multiple evidence have shown that these mutations associated with immune escape. This variant causes higher viral load, which may associate with its higher transmissibility. There's still no evidence indicating this variant being associated with more severe disease or worse outcome. But it did demonstrate worrisome immune escape in vitro study and also in the clinical trial. So I will show some data later on. The Brazil variant, another worrisome variant, was first reported in Japan for travelers from Brazil during a routine screening at the airport. It contained K417T, E4A4K, and N501Y mutations in its spike protein RBD, receptor binding domain, exactly the positions where 3 mutations were found in the South Africa variant. There are evidence suggested that the mutations may affect the ability of antibodies generated in previous natural infection or through the vaccination to recognize and neutralize the virus. The resurgence of COVID-19 in Brazil town Manaus provided the evidence showing how the Brazil variant attribute to the immune escape. The top panel of this diagram here showed the number of daily hospitalization and deaths in the town. You can see after the first wave of pandemic, the number remained stable and fairly low for 7 months from May to November. Studies have indicated that approximately 75 people have been infected by the SARS-CoV-2 as of October 2020. This percentage is beyond the herd immunity threshold, which is 67%. You may think people in the town have been protected well. Unfortunately, unexpected abrupt increase of number of daily hospitalization occurred in early January this year. 42% of the infection was identified as Brazil variant in this region from December. In contrast, this variant was absent in samples analyzed from March to November. This suggested there's a strong correlation between resurgence of COVID-19 and the explosion of Brazil variant in this town. Then we talk about the vaccines. To bring the pandemic under control, massive global efforts have been put on the development of antiviral drugs, therapeutic antibodies, and effective vaccines. Vaccination is still the most effective way to reduce the spread of virus and prevent the infection. Current vaccines all target the entry step of virus infection by blocking the spiking protein binding to its receptor ACE2 on the host cell membrane. During the pandemic, the vaccine were developed and approved at unprecedented speed. There are wide varieties of approaches to develop the vaccines. Here we have the inactivated or attenuated virus vaccines that were pursued by several Chinese companies. 2 of the vaccines have been approved in China and some other countries. Also, we have the recombinant protein or peptide protein vaccines. The Novavax just released their clinical trial result of their protein vaccine recently. It has impressive efficacy. For engineered viral vector vaccine, predominantly the adenovirus based, we have vaccines from J&J and AstraZeneca. Both vaccines were approved with efficacy around 75%. Then we have RNA vaccines. That is something never done before. The mRNA vaccines developed by Moderna and Pfizer BioNTech have been approved with great efficacy. Here is the data on Moderna and Pfizer clinical results. The 2 vaccines have a very similar effectiveness. Shortly after the first dose, even before the second dose, the curves start being apart. Number of cases in placebo keep rising. The number in the vaccinated group stays relatively flat. The overall efficacy in protection of mild to murder rate COVID-19 is 94.1% for the Moderna vaccine and 95% for the Pfizer vaccine. Scientists knew the SARS-CoV-2 may evolve to escape immune response. So from the beginning of the pandemic, they have started to conduct various experiments to try to understand the pattern of emergence of the escape mutants. A research group from Washington University engineered a pseudovirus with a membrane expression of SARS-CoV-2 spike protein. They used the pseudovirus to infect the cells in the presence of monoclonal neutralizing antibodies. The virus that evaded the antibody neutralization and kept infecting cells were isolated and sequenced. From this experiment, they found the variants with mutation at E4A4K or A and F4A6S. Actually, the Brazil and the South Africa variants has this E4A4K mutation. Then they performed the same experiments with 9 more neutralizing monoclonal antibody. 29 identified escaped mutants were tested for the degree of resistance to the neutralization by different monoclonal antibodies. The levels of resistance were represented as a heat map here. From white, the low degree, to the red, the high degree. Most of the mutations were associated with resistance to more than one monoclonal antibody. Mutations at S477 and E4A4 showed a more broader assistance. Actually, S477N was later detected in a variant quickly spreading in New York City, and E4A4 mutations were shared among the several escape variants in the circulation. The escape mutants were tested in the neutralizing assays with a full convalescent sera. In the heat map in the middle, the neutralizing activity was represented by the color shade from white, low activity, to blue, the higher activity. It is reviewed the mutation with substitutions at residue E4A4 were resistant to each of 4 sera samples. That suggests that this is a dominant neutralizing epitope. To understand how SARS-CoV-2 can adapt to evade the neutralizing antibody, a research group from Rockefeller University generated a chimeric virus with expression of SARS-CoV-2 spike protein and the green fluorescent protein as a reporter. They used the chimeric virus to infect the cells with or without the neutralizing antibodies in the culture. Without antibodies, the cells were infected and have expression of GFP. However, in the presence of antibodies, the infectious unit estimated by the frequency of GFP positive cells were significantly reduced. After serial passages, the culture supernatant was collected and the viral RNA was extracted. Sequence encoding the spike protein were amplified and sequenced. The sequence analysis reviewed a clear selection of the variants under the antibody selection pressure. Similar or identical mutations emerged in the presence of each of 3 monoclonal antibodies. In the case of antibody C121 and C144, mutations E4A4K and Q493R or K had a higher frequency in the virus population. Higher frequency of the population, higher frequency was observed in of the mutations at R346 and R440 in the virus population passed in the presence of antibody C135. All three antibodies can inhibit the infection of white chimeric virus with a high potency. However, the mutations in the spike protein like Q439R result in the complete loss of neutralizing activities of monoclonal antibody C121 and C144, while the mutation R346 had a similar impact on the monoclonal antibody C135. Actually, when the Rockefeller group first discovered this E4A4K escape mutations, it has not been detected in circulation in the natural world. Now more and more evidence suggesting this key mutation attributed to the antibody resistance. Both South Africa and the Brazil variant have this mutation. The original UK variant does not have this mutation, but just recently it was reported that this mutation was found in a new derivative UK variant. This is alarming because we know the UK variant has a higher infectivity and transmissibility. Now with the addition of this immune escape mutation, the variant becomes more dangerous. On the right side of the slide, it is the top view of a functional spike protein, which is homotrimer. This is the surface where the spike protein bind to the ACE2 and some neutralizing antibodies. The notable mutation, the Q mutations in those variants of concern are all around this area, the region. The mutations make it difficult for the antibody to recognize and bind to the spike protein. Later on, the group isolated more neutralizing monoclonal antibodies from the individuals who received the vaccines. They performed the similar experiments with monoclonal antibody to study the emergence of escape mutations on the antibody selection pressure. After replication in the presence of each individual monoclonal antibody shown on the left side, the viral sequences encoding spike protein were analyzed. This table showed the percentage of viral sequence with indicated mutations identified from the variants coming out from this selection experiment. The mutations are showed across the top. They found the mutations at K417, exactly the position where we found the mutations in the South Africa and the Brazil variant. The N439K is the variant that achieved a significant number of cases in Scotland initially and later in parts of Western Europe. The N440 are variants that achieved a higher prevalence in India. L452R is so-called the California variant, which raised quite concern in Southern California area. Next two mutations, F456 and N460, have not hit the news yet, but I believe they will appear in the future. Again, we have a E4A4 mutation. It seems a number of antibodies selected this variant in the selection experiment. Then there are two more variants that has not been hit the news yet. And after that, we have N501Y, the Q mutations in all three variants I'm talking about here today. Since the outbreak of COVID-19, we at Signalchem took quick action in response to addressing the needs for developing effective therapeutics and diagnostic of COVID-19. One of our projects was to co-found the Canadian Coalition for COVID-19 with several Canadian companies. Its strategy goal is to generate nanobodies for accelerating development of tests and treatments of COVID-19 by sharing the sequence in open source model. We provided our recombinant SARS-CoV-2 RBD protein to immunize the llama, and later on a phage display library was generated representing the antibodies elicited in the immunized llama. After several rounds of screening, 51 specific nanobodies were obtained. We produced the recombinant nanobodies in E. coli and CHO cells and performed characterization studies. It was revealed that 3 of the nanobodies demonstrate a high potency in inhibiting RBD binding to soluble ACE2. Those nanobodies also block the spike protein binding to ACE2 receptors expressed on the cell membrane, which is a critical step for the virus to gain the entry into the host cells. That means these nanobodies have the potential to neutralize the SARS-CoV-2 and provide the prevention and the treatment of COVID-19. Since the new SARS-CoV-2 variants were reported, we produced a recombined spike protein or its RBD carrying the mutations identified in the variants. Then we tested the inhibitory activity of our nanobodies against those RBD mutants binding to the ACE2. The results showed that N501Y, the K mutation in UK, Brazil, and the South Africa variants, had a minimum impact on the inhibitory activity of a nanobody 20014 and 20038, but completely impaired the activity of nanobody 20025. The RBD mutations in South Africa variant K417N, E484K, and N501Y caused a remarkable decrease of inhibitory activity of all 3 neutralizing nanobodies shown here. This confirmed the finding that the triple mutation in South Africa variants attenuated the neutralizing activity of antibodies. All the data I just present here indicate that in vitro study, the experiments have done a pretty good job to predict and define the emergence of antibody neutralizing escape variants in the natural world. Then we will ask ourselves a question: what impact do new variants of COVID-19 virus have on the vaccines and therapeutic antibodies? In this study, neutralizing activity of sera from Moderna mRNA vaccine phase 1 trial participants were measured with pseudovirus incorporated mutations present in the UK or the South Africa variants. The key mutation or the complete set of mutations of UK variant had a minimum effect on the neutralization of vaccinia sera. However, a significant decrease of neutralizing activity was observed against the virus with a full set of mutations present in the South Africa variant. Even the combination of 3 key mutations in RBD already reduced the sera neutralizing activity. Recently, Pfizer published their research data on the neutralizing activity of its mRNA vaccine elicited sera against the UK, South Africa, and Brazil variants. Neutralization of UK and Brazil variants was roughly equivalent compared to the early strains. Neutralization of South Africa variant was significantly lower, especially the one with a full set of the mutations. The research group from Columbia University investigated the impact of mutations presented in the UK and South Africa variants on the SARS-CoV-2 monoclonal antibodies. They studied the effect with authentic virus and pseudovirus containing individual or combined the mutations. The top panel showed the top view of spike protein trimer and the different side view displaying the binding sites for the 12 monoclonal antibodies tested in this study. For neutralization of a UK variant, only the activity of 2 antibodies are significantly impaired. However, for neutralization of South Africa variant, the activities of 5 monoclonal antibodies are completely or markedly impaired. Those antibodies include antibody from Eli Lilly and Regeneron, though those antibodies have been approved for clinical use. This table here summarizing the changes in the neutralizing IC50 titers relative to the Y type. The red color represented any changes greater than 3-fold. It is clearly indicated that the K417N and especially the E4A4K played a critical role in the neutralization resistance. The group assessed the neutralizing activity of 6 monoclonal antibody that bind to the spike protein N-terminal domain and tested their activity against the 3 UK and South Africa variants. Both UK and South Africa virus are profoundly resistant to 3 antibodies. The activity of other 3 antibodies are variably impaired, particularly against the South Africa virus. They also examined the neutralizing activity of monoclonal antibody therapies in clinical use or still under the clinical investigation. Notably, the Eli Lilly antibody, the commercial antibody COV55, alone or in combination, is no longer able to neutralize South Africa virus. The combination of Regeneron 2 monoclonal antibody approved monoclonal antibodies shows noticeable decrease in its activity against the South Africa virus as well. These findings suggest that antibody treatment of this virus might need to be modified in the localities where the South Africa variants and the related variants are prevalent. And it is important to use a combination antibody therapy to address the neutralization resistance of emerging variants. Neutralizing activity of 20 convalescent plasma were evaluated against UK and South Africa variants. 16 of 20 plasma samples lost greater than 2.5-fold neutralizing activity against the South Africa variant, while still maintain activity against the UK variant. Sera were collected from individuals who received the Moderna or Pfizer mRNA vaccines. The neutralizing activity of each vaccine serum samples was assessed against the UK and the South Africa variants. The results shown here, no loss of neutralizing activity against the UK variant, whilst every sample lost the activity against the South Africa variant. So findings in these two studies, as others are reporting, raised the concern that the new emerging variants are threatening the efficacy of monoclonal antibody therapies and the vaccine protection. Then how do the new variants affect the effectiveness of vaccine in the real world? We can learn some answer from the clinical trials. First is the AstraZeneca vaccine. It is a viral vector-based vaccine. 11,636 people participated in a phase 3 clinical trial in UK and Brazil. The participants received 2 doses 28 days apart. The vaccine is a 76% efficacy at preventing the COVID-19. So it was approved in UK, Brazil, and some other countries, and EUA are also granted by EU and the WHO. In South Africa, the clinical trial participants received 2 doses spaced 21 to 35 days apart. Unfortunately, the protection efficacy was only 21.9%, far below the 50% minimum required for EUA in many countries. As a result, the South Africa government suspended the use of this vaccine in the country. Researchers sequenced the virus that infected the trial participants and found a strong link between the vaccine failure and the explosion of South Africa variant. Recently, this vaccine is suspended in more countries due to blood clot concern. Now we have the Novavax vaccine. This is a protein-based vaccine. 15,000 people participated in the clinical trial in UK. They all received 2 doses of this protein vaccine. The recently released clinical data showed this vaccine delivered a higher efficacy at 89.3%. 50% of the infections were highly transmissible UK variants. Actually, for preventing the original strain of virus, it showed efficacy at 96.4%. It's a pretty high efficacy. It also proved 100% effective against severe disease and death in the UK study. So according to the clinical result showing here, I believe this vaccine will be soon approved by countries. 4,400 people participated in the clinical trial in South Africa. Among the participants without the HIV infection, the efficacy is 60.1%, including those individuals with infected HIV. The efficacy dropped to below 50%, 49.4%. 92% infection was South Africa variant. So apparently, the South Africa variant caused a decrease of protection efficacy in South Africa. The Johnson & Johnson vaccine is also a viral vector-based vaccine. Clinical trial participants were from US, Central and South America, and South Africa. All participants received only 1 dose of vaccine. The overall protection against moderate to severe COVID-19 infection was 72% in the United States, 66% in Latin America, and only 57% in South Africa. I believe the lower efficacy in Latin America and South Africa was associated with the Brazil and the South Africa variants being circulating in those areas. The vaccine have been approved by the US, Canada, and other countries, and the WHO also listed as for the EUA. The vaccine demonstrated 85% efficacy against the COVID-19 related fatalities and severe symptoms in all age groups, regardless of the person's other underlying health conditions. It also provided 100% protection when only considering the deaths or the COVID-19 cases that required a hospitalization. Now we see the emergence of new variants and their threats to the efficacy of current antibody therapies and vaccines. And it is believed that new escape variants could emerge in the future and lead to a severe epidemic rebound. Then what should we do and what can we do? First, we need to do everything possible, including the non-pharmaceutical intervention and scale-up vaccination to reduce the circulation of the virus, to prevent the infections and reduce the opportunity for the SARS-CoV-2 to evolve, resulting the mutations that may reduce the efficacy of the existing vaccines and monoclonal antibody therapies. Then we need to continue to conduct the surveillance of the circulation of SARS-CoV-2 variants through testing and sequencing, and collecting the samples of sera and cells from individuals with past infection or past immunization with available COVID-19 vaccines for serum neutralizing and cellular immunity functional testing against the newly discovered variants that can provide the guidance to the minimum set of epitopes that are required in the new vaccines. The production of COVID-19 vaccines has to be reactive and to adapt to match the new emerging variants in the circulation. Finally, we need to ensure global access, availability and affordability of COVID-19 vaccine to ensure no countries are left behind. The end of the pandemic is only possible when the effective vaccines against the circulating variants can be distributed equitably and across the world. So the emergence of SARS-CoV-2 variants, especially those variants have a potential immune escape to the antibodies, pose the challenge to the efforts for ending the pandemic. But with the increased knowledge of the disease, the virus, and also the successful development of the vaccines, the current effective vaccines, I'm pretty optimistic that we are going to develop and have a more preventative or the therapeutic approaches to have a good control of this pandemic. Okay, so thank you very much for your time, for your listening.
Thank you, Clark, for that very informative and educational webinar. For those of you who have not already, please feel free to use the question and answer portal to post any questions to Clark so he can try and address them in this webinar. As we already have some, I'm going to move straight into some of those questions, and Clark, feel free to comment. The first question is, the in vitro work in escape mutations seems to suggest that the virus evolves at an alarming rate. What are your thoughts on the effect this has on herd immunity?
Yeah, this is a very interesting and important question. Given the emergence of variants with higher infectivity, transmissibility, and the potential immune escape to the antibodies, as I just showed, no doubt it is getting tougher and tougher to achieve the herd immunity, but there's still a hope. How many people will need to be vaccinated to stop the disease spreading? This depended on how infectious the viral strain is, how the effective the vaccine is, and also how much we prepare to continue with the lockdown measures. We know people have been tired of those lockdown measures and try to relax it, but we need to keep in mind it is also associated with the vaccination level needed for the herd immunity. The model prediction for the success of the vaccine program depends on the vaccine being able to limit the transmission of the virus, not just to stop the people getting ill. But we still don't know. We still don't know if the current batch of the vaccine can stop the transmission. The good vaccine has to be able to not only protect the vaccinee, but also protect the people around the vaccinee. Because in the clinical trial, only the symptomatic cases were counted for the efficacy assessment. And also, we still do not know for sure how fast the new strain can spread if allowed to do without any mitigation measures. For those highly transmissible variants like UK variant, we know that even higher vaccination levels is required to achieve the herd immunity. But on the positive side, a previous COVID-19 infection still provided a good protection against the reinfections. Actually, the reinfections are still rare. So we can probably count also on nearly about 20% of the population that having the natural immunity. And anyway, to achieve the herd immunity, I think we need to have a persistent long-term plan, including both quickly rolling out the effective vaccines and continue the social distancing and testing.
Thank you, Clark. Question 2. Given the surge in variants that has occurred, and no doubt will continue to, should we as a community be more focused on better viral treatments rather than preventative measures such as vaccines?
Yeah, I think my answer is that we need both. We need both. We need the antiviral drugs even when the vaccine works very well. The reason is not everybody has a strong enough response to the vaccination or vaccines, and not everyone can or will want to be vaccinated. Some people don't like the vaccination. Moreover, now we see the new variants that can impair the efficacy of current vaccines, right? For a case in which the immune response cannot provide effective protection, we definitely need antiviral drugs to treat the patients, just to avert the progress to a severe illness and eventually death. Mass vaccination program is still the most effective way to reduce the spreading of the virus and prevent the infection. Current vaccines work pretty well to protect the mild to moderate COVID-19. Although, as I showed, some escape variants can attenuate the vaccine efficacy to a certain extent. But the vaccine do shows good protection, very effective protection against the severe disease, the hospitalization and death. You know, this will help significantly to ease the challenge, the burden on the public health system during the pandemic. So my answer is we need both.
Thanks, Clark. Question 3. Do you have any more ideas into treatment options, antibody, protein, or small molecules?
Yeah. There are various options for the treatment even now, but the research all over the world are still working very hard to develop more treatments with different strategies. So I just give some samples here. The first of all, of course, the therapeutic monoclonal antibodies. The antibody can interfere the binding of a spike protein to ACE2 receptor, the critical step for the virus get into the cells. The blocking can to lead to the inhibition of the infection. 3 monoclonal antibodies have been granted with the EUA, 1 from the Eli Lilly, 2 from the Regeneron. But as I just showed in my presentation, all 3 monoclonal antibodies was impaired by especially the South Africa virus. So updated monoclonal antibodies need to be developed. And according to the data I just showed, I think in the future, the antibody cocktail, the combining antibody use for therapy is the way to treat the COVID-19, to address the emergence of the escape variants. Secondly, we have people are developing the soluble human ACE2, the receptor decoy, that can bind to the viral spike protein to block its interaction with ACE2 on the membrane of host cells and inhibit the viral infection. Clinical trials are ongoing. Actually, at Signalchem, we are collaborating with Austria company Innofo to use their unique AI platform to develop more potent soluble ACE2 for the COVID-19 treatment. Then we have the antiviral drugs. Those drugs are aimed at protein targets that are important for viral life cycles, like proteases and polymerase, to inhibit the viral replication. You know, the remdesivir, Gilead compound, a SARS virus RDRP inhibitor, has been approved to treat the COVID-19. As I just showed in my presentation, actually there are other membrane-associated receptor also play a role in the virus entry to the cells. So like the AXL is the one of the example. So blocking or inactivating of AXL showing the efficacy to inhibit the infections. We have at Signalchem, we have a very potent and selective AXL inhibitors from our oncology program. Now the compound is in the clinical trial. So we just take advantage to develop this compound as anti-COVID-19 therapy. It's in the progress. Lastly, we can use anti-inflammatory drugs. Some patients may develop a hyperimmune response, so-called cytokine storm, to the viral infection. In these cases, it is the immune system's overreaction that is damaging the lungs and other organs, and too often leading to the death. So it makes biological sense to treat the patients with inflammation suppressor, like some of the corticosteroid, to minimize the risk of immune overreactions. Yeah, so those are some options for the treatment of COVID-19.
Thank you, Clark. That was a very informative answer. Unfortunately, we've actually reached the end of our webinar time slot here. For those whose questions have not been answered, either provide your email address in the private reply, or please try to contact Clark at info@signalchem.com. Thank you everybody for attending this webinar, and we hope to see you soon.
Thank you, everybody. Thank you.